So let's move on to the second talk, which is the domain shift and interspecies transfer, given by Marc Aubreville. Marc is the team head and professor for image understanding and medical application of AI at Technics Hochschule in Großstadt. Sorry for pronunciation.
Very good.
PhD from Friedrich Alexander Universität Erlangen, Nuremberg, and his MS from the Karlsruhe Institute of Technology. Marc's vision is to harness the power of AI algorithms and apply them to clinical applications.
Yes, and I would love to share my screen, but unfortunately, I think someone has to declare me a host first.
Okay, we'll do it. How can I do it?
You just call it quickly. We've already made Jim down the host.
Sorry, you have
Ask
to
Jim down to make you the host.
Oh, yeah.
Yeah.
That's all right.
No, not yet. Hold on.
Don't make host. Just allow sharing of the screen by multiple people. Most of them. Don't make host.
I mean, I think you can. You should be able. Yeah.
Now. Yeah.
I am now.
All right.
Perfect.
Yes,
so then I would be ready to start. So, I mean, first of all, thanks a lot for having me on this very interesting workshop that you have put together on such short notice. I know, and thanks for the host of the session for introducing me. So I know that German names are so hard. So kudos for trying it, and thanks anyway. So my talk today will be about domain shift and interspecies transfer, and this sounds maybe a little bit like an odd topic, and I think I will motivate the rationale behind it, what we're aiming to do, and the steps so far that we've achieved. Maybe I should also highlight that we recently launched a kind of collaborative group called Deep Microscopy, where basically most of our research is now also happening on a website. It's done together with Katharina Breininger. Short shout out to her from FAU in Germany, and I think it's very nice. So let me get into the talk. And as I know that you record this talk also for YouTube and so on, I might briefly introduce like what is domain shift, domain generalization about. For most of you, this will be of course already known, but just to give you a nice example here. So I think the people at the University of Warwick know maybe this scene here. It's actually from very close by. It's the Trinity Street in Coventry, and it is actually, from my perspective, a kind of nice example on what we're also trying to achieve, like here now as a real world example. So here on the left hand side, you of course have like a mid 1950s photography of that very same house and scene, and then in 2012 on the right hand side, of course, it looks all very different. Now, while you could say, oh, well, okay, so the domain shift is obviously just a black and white photography now to a color photography. Of course, it's not. So there's one thing that is of interest. Maybe it's this house. And then you have like multiple things around it that have changed over time. And this is something that we are very familiar with also, like in histopathology, that we are interested in a certain thing, and this certain thing will change actually over the course of like being in a different environment, being in a different stain, being in a different whatever, right? So it's, I think, a very nice example of that. So, I mean, the problem in general, I think it's clear for everyone, just repeating it. So while we might have a maybe even more subtle domain shift in the input of our systems, so from the source domain, which is what we train on, to the target domain, which is what we want to apply our systems on, after running it through our network, for example, CNN, we will have a typically even larger representation shift, which will make our task, a classification or whatever, more difficult. Now, I started off in the first slide by saying we want to do interspecies transfer, and then you might ask yourself, so why is that, and why is it even important? And I will also come to the question of, is it even necessary? But before we do that, let me just briefly motivate on why we are like in the working on the intersection of human and veterinary pathology. So of course, it's, I think, known to everyone that animals have successfully been used as model for human diseases. Sorry, the typo there on the slide. And also, animals do have spontaneous tumors, so not like specifically induced tumors. And we find this, of course, also in pet animals where it's being treated regularly. And we also find great similarities in the pathogenesis, in the tumor pathology, and also in the treatment options of those. So it makes a lot of sense to join forces here also with like the veterinarians that are working on this topic. And I mean, this is one thing that for me as a machine learning scientist is also very beneficial. The regulations governing animal specimens are significantly less stringent, right? So we can actually use this without any kind of ethics violations, ethics boards approval, and so on, because this is typically, I mean, animals are considered a thing by law, so they have no personal rights, and especially if we're like doing stuff that we usually do, like we're taking some samples from the diagnostic archive, which is actually not an ethical problem at all, then we are also out of the regulations. And the reason why we are so confident about like working on animal specimen is actually because like when we started this like several years back, I talked to Christoph Bertram, who is a good collaborator of us. He's at the veterinary medicine at the University of Vienna. University of Veterinary Medicine in Vienna, and he told me that like from the microscopy image alone, I can hardly tell if I'm looking at a monkey or a guinea pig. So that gave us a clue, because I mean, if there is something that is so subtle that for a human it's hard to tell the difference, maybe also the domain shift is not that severe. So the research questions that I'm trying to like answer, or maybe not to raise at first in my talk, are like, what are the main sources of domain shift in histopathology? Are humans also susceptible to domain shift? And if yes, then how much? And well, of course, the ultimate question, like, how do we make models robust against domain shift? Now, the last question, I can already tell that, I mean, I won't have time to dig too much into that, and I think there's also like plenty of literature about that already. But let's now go for the first question at first, and which is like, which are the main sources of domain shift in histopathology? And if you look at older papers, then you will find that they will typically say, oh well, you know, it's mostly the tissue processing, because the tissue processing is different at every lab, and that means that, you know, like, because of different tissue processing at every lab, we have those huge differences that all of us are able to observe. I mean, long story short, we already know that the tissue processing might actually not be even the worst of the influencing factors. Another one that might be of value is the species, which we will tap into briefly into this talk. The imaging device, which we already know has a significant impact, and we will also talk a little bit about the MIDOC challenge, which actually tapped into that. And then finally, the tissue type, so like which kind of tumor in which kind of species, and so on. So the question here is, which are the most dominant sources of domain shift? And this is, of course, the one that we would like to like then work on and make the whole system robust against. Right, so let's start with this. Before we do that, just like, let me note that, of course, it really depends on the task, right? So you have some tasks, like, for example, tissue segmentation, where our PhD student, Frauke Wilm, has done a great deal of work on, where you know that, well, actually you have a kind of low dependency on the color contrast in the recognition. You have a high dependency on the morphology, because like tissue segmentation is mostly about morphology. And you have a high dependency, of course, on the context. Then you have other things like the IHC cell detection, right? Immune histochemistry cell detection, where you have a specific stain, and a specific stain is what you are actually looking for. In this case, you have a high dependency on the color contrast. You cannot just like change the hue, for example, of this image. It will just not work anymore. You have to be careful when augmenting these images, and so on. On the other hand, you have a rather low dependency on the morphology, because you have the specific stain that highlights everything that you want to have, and you have a low dependency on the context also, right? Because also the context is not that visible anymore. And then we have my prime example. I don't know, it's just one of many examples, but it's an example that we can, of course, use because it's an especially challenging task, the task of mitotic figures. So mitotic figures are cells undergoing cell division, as most of you will know, and they're now highlighted in this image, and they have actually a particularly high dependency on the color contrast because it's really, they need to be dark blue and not black, for example. Black would rather be like staining, error staining particles, but they can have a very similar morphology. You are additionally dependent on the morphology in order to differentiate against some granulocytes, for example. And that would be one thing. And then you have also a high dependency on the context to see if there's actually like tumor tissue around, right? So then it wouldn't really matter. So while we don't have time for everything, let's briefly dig into this topic, which I think is like one of the most challenging, and it's also one of where we have like most experience in and can tell a brief story about. So first up, topic of tissue preparation and species. And already I have to tell that, I mean, this is really hard to disentangle because you typically have like labs that are working with human tissue and labs that are working with veterinary tissue. So you cannot really disentangle those really well because you typically don't transport the tissue to another lab and have it stained and processed there, right? So this is typically what you don't do. One of the results that we achieved in this is that we took some like dog tissue from a data set that we actually published, which is here on the left-hand side of the screen. Hold on, let me get my pointer there here. And then we compared this and ran inference on the AMIDA13 dataset, which is a publicly available dataset. Now, interestingly, both use a very similar scanner. So the color impression, the field, depth of field, and so on is very similar. And we also see this in the results. So if we run like a just default RetinaNet object detector, we can see that actually, well, there is a drop in performance, like coming from the canine data set to the human data set, like here, right? There is a drop in performance compared to training in domain and evaluating in domain, but it's not a very strong drop. And of course, there's like lots and lots of different influencing factors that could also be part of this. Like we could have, for example, also a small possible shift because it wasn't the exact same scanner, also like the tissue processing and so on. But we could just say, you know, like it's not a major influence. Small asterisk here is that both data sets used a rather similar tissue preparation, although they come from like entirely different labs, right? So this is just what we can see visually. Next up, let me talk about the influence of the digitization device. And this is something that was underrated for a long time because like people just assumed that it would be the tissue preparation that gave this huge differences in the staining. And if we look, for example, at this image digitized with I believe it was a Hamamatsu XR scanner, and then you have the same, very same slide digitized with another scanner, then you can also see that it's a strong difference in like contrast, in of course the hue, and so on. So there's actually a lot of problems associated with it, and it leads to like a really strong drop in performance, as we were able to show like in this table. So you can see that if you go like from the same device here, and here, then you have a much stronger performance than if you go like cross device without like any specific augmentations against that. So this kind of laid down the foundation and the argumentation for the MyDoc challenge, which we then ran on MyDoc on MICAI in 2021. So this is the mitosis domain generalization challenge that we ran there. We had a training set using like 4 scanners. It was all from the same pathology archive from UMC in Utrecht, in the Netherlands. So a short shout out to those guys who helped us a lot running this whole thing. And then we had another set of scanners. So 2 were actually even part of the training set. So this one here and this one here, while this one was, however, not labeled. So we only had some images part of the training set. On the test set, however, we had also 2 additionally completely novel scanners, and we wanted to see like how well does this all generalize. Just to report about the challenge setup, which is I think something that especially I'm expecting that we will see some challenges on domain generalization in histopathology in the next years. So maybe this is some things that we think is good that we could recommend to other people. So first of all, I want to highlight that no images of the test set were ever made available to any participant, right? So while you would say that this is common sense, for many challenges, the images are in fact available and only the labels of the test set are hidden. So we didn't make that available to anyone and it's still secret. Then the participants were required to publish a preprint about their approach, which is also something that we can really recommend to any organizer of a challenge, because it really helps you to summarize everything at the end. And the ground truth we got was by an object level agreement of 3 experts, which is of course also, well, from my point of view, really good because it really reduces the labeling noise that you have. I'll talk briefly about that on the next slide. It was the first challenge to investigate the domain generalization in histopathology, at least from what we know. It was the first challenge to really require a Docker container submission on Grand Challenge org for that purpose. So we thought that this was a big success. Also, the team from TIA participated with some success, no, with a lot of success actually, in the challenge, in both actually challenges. And the next one I will talk about a little bit later. So just briefly, how did we make sure to keep down the labeling noise? Because mitosis is something that is very prone to labeling noises. Indicating that we had one pathologist actually looking for true positives or like real mitotic figures, and also some non-mitotic figures, something that looks very similar. We handed this over class blinded to a secondary expert, and if both experts agreed, then we added this as a positive label. If they disagreed, then we had a 3rd expert rated. So we have always a majority of 3 experts, because if 2 already agree, then the 3rd doesn't need to be asked for that, of course. We additionally had, because mitotic figures are so easy to overlook, we had additionally a machine learning algorithm screened for additionally maybe just some cells that were overlooked, and for that we also ran those without any kind of labeling through the experts, and looked if they agreed, and if not, then we had a 3rd expert looking into this. So this is would of course not have been possible without the great help of Taryn, Christoph, and Robert, who made this. So a shout out to them here. One of the questions that we've been asked over and over again is something that I haven't talked so far about, but it's, I think, very interesting. So since we're working with the same tissue, why not just annotate on one slide and then register to all scanners, right? That would be actually much easier for us to do. And the answer is actually not as simple as you would expect, because so we did this kind of experiment post hoc, post the challenges, and we found out that rescanning a slide, so even maybe with the same scanner, leads to very different settings for the fine focus on an image region. And that means that depending on the scanner and so on, you could have up to 10% of the mitotic figures now being totally blurry, like shown up here, so undistinguishable blurry. And we have to assume that this problem is actually even symmetric. So meaning that in our original data set, we also have 10% missing that would then suddenly show up if we scan it again, right? So this is a big problem, and this is why we didn't really do that. Now, let me just briefly come to the results. I think this is not really so important, but of course, it was interesting to see which kinds of approaches were used. So we found that some of the participants used a cascading approach, a multi-stage approach. Some used ensembling, test time augmentation. We were actually surprised how little or how few of them used that, because it's a very common technique for challenges to do ensembling, right, of models. Then we had a lot of people using stain normalization. We had other people doing domain adversarial in the various form using generative adversarial networks and so on. And then we also had domain augmentation as a different technique. And finally, we had some people that just said, well, while this is actually an object detection task, we will just interpret it as a kind of segmentation approach, be it an instant segmentation or a normal semantic segmentation approach with some post-processing. Now, if you look at that, then it would seem that, oh, well, apparently the instant segmentation is the one ingredient that made this all really, really good. On the other hand, after having some experience with that, we also tapped into that, and we found that maybe it was just the teams that worked with instant segmentation were also the teams that had a really, really good engineering done on the task, right? So they really did a good job. We can also find that results across scanners still varied by quite a bit. So these are all like of them on the known scanner, the one that was part of the training set. If we then also enable like the other one, which was part of the training set without any labels, then we see that this already varies by quite a bit across participants. Now adding the unknown scanners, first the 3-day HISTEC one, and then another one from Hamamatsu, we can see that it's actually quite a spread in performance. So this is all the result of bootstrapping, and you can see here nicely that actually the group that won, or the groups that won, they had a pretty good performance across all scanners, right? And this is of course what really makes a generalization. Another thing that is really worth talking about is actually the comparison across the systems. We have here a precision and recall plot, and we see that actually all of them did a pretty good job in finding a proper operating point of the algorithm, right? So it's a pretty good and decent precision and recall trade-off. Now, also, we see that the inherent dataset diversity in the training set, it really already strongly helped the models to generalize to the scanner domain, rather independent of the actual approach that was used. So we had no complete failures in the challenge. What is also really worth digging into is the question of like, how good do human experts perform? And this is like part of our medical image analysis media paper that we then published a couple of months later. So we had actually some experts do the same kind of tasks, and we compared also like precision and recall on the challenge ground truth and also on an independent ground truth that we then evaluated again. And we found that basically we could say it was a little bit all over the place. So you had persons with a very high recall, you had persons with a very high precision, you had somebody in between. So I mean, this was a very limited sample of experts, but it still tells us that actually maybe the human kind of performance is overrated in this kind of task, which makes it a task really worth optimizing. All right, another thing that was very interesting for us, and shout out to Katharina Breininger who did this plot here, is like how dependent are the experts actually on the imaging device? Like, because we always assume that experts are not dependent on the imaging device, but we found out that yes, in fact, they are also dependent. So if you look at this here, down here, this is now, so always like such a spider here is one expert, and this red points here is one scanner. So you can see that for one scanner, basically just performance really dropped. But for other scanners, for example, the scanner here in blue, we see that some people have a higher precision, whilst others have a higher recall. So we could just say it's not a general thing, right? So there is a strong dependency on the scanner, even for humans, which is really an interesting finding from our perspective. Yeah, so that was kind of the first three categories that I wanted to talk about, like imaging device. And finally, we want to talk about the tissue type. We just ran some experiments and that were just accepted by scientific data. It's not yet available, the DOI that you see here on top, but it will be very soon, hopefully, if Springer does their job quickly. What you can see here is that if we train on like some kind of tumor domains, like neuroendocrine tumor, lymphosarcoma, lung carcinoma, and so on, and we test this against an independent set, which was here the MIDOC2022 test set, actually, the successor of the 2021 challenge, then you can see that it's also varying very, very strongly. So training on one tumor and tissue type will not generally generalize well to other tissue and tumor types. Especially if you look into the details, you find some surprising results. So for example, you could say, okay, so if you train on breast carcinoma, lung carcinoma, then this also works pretty well on bladder carcinoma. Yeah, it's all carcinoma, so that makes a lot of sense. But then if you look here for another data set of memory carcinoma, actually also breast cancer in dogs, then you find, oops, the performance drops all of a sudden. This is all mean values out of 5 runs, so it's a pretty solid finding. On the other hand, if you train with multiple domains, so all of the domains here, then you can find that we have a pretty solid performance across domains, which also tells a story. So I already teased the MIDOC 2022 challenge, which we then ran last year. which was also really awesome. And we there used no longer like different scanner types, but different tumor types. So we had 6 tumor types in the training and independent 10 tumor types in the test. And of course, this also includes differences in tissue processing, species, and scanners, and so on. And what we found there is that if you train, for example, only on breast cancer, which was our like baseline approach from the last year, if you compare that to something trained on 6 tumor types, some 6 tumor types and adversarial, and like the winner of the challenge, which was also the TIA team actually, then you find that there's a strong kind of a benefit that you have from including the data diversity by itself. So take home is data diversity is actually what you need, right? So attention, okay, but also data diversity is what you need in your data sets. And if you would ever have the chance to increase your data diversity, do that. We also did that because we published the MyDoc22 challenge dataset in an extended version, which we call MyDoc++. It was just accepted, as I told, in Scientific Data, and should be out there like any day now. In total, we have like 503 cases, some 12,000 mitotic figures, some new tumor types, and so on. So just include more diversity into the dataset, because ultimately, as we have learned from like large language models, of course, as well, data diversity is key. So let me summarize a little bit our findings. So from our so far investigations, we would say that the species is the least of the sources of domain shift, followed by the tissue processing, like inter and intra-lab differences, of course, followed by the imaging device, and finally the tissue type. Why? Because the tissue processing is basically impacting the color and the staining, maybe also the tissue thickness, which is also like an important source of domain shift. While the imaging device, it impacts the color representation, the depth of field, which is often like ignored, and the tissue type impacts basically everything, the complete structure. Now, I told you already that diversity is key for these kinds of things. And I forgot to mention one thing. And the one thing is that we were typically only looking at like regions of interest so far. And the regions of interest, they also only tell us like part of the truth. And this is something that we found already in an earlier publication of ours. So if you enlarge the region of interest from the usual 10 high power fields to 50 high power fields, or to even the complete slide for training, then you also get a significant boost in performance also on the complete slide. So this is something which is, of course, also has to do with generalization, because you suddenly have like cell types, maybe like dying cells, necrosis, and so on, that were not part of the original data set, and that now will hurt your detections on the new data set. Yeah, surprisingly, on the same data set, we also evaluated like how many whole slide images do you need, and we found a much lesser dependency there in terms of then to the F1 score. All right, I want to close basically with one kind of case study that kind of shows that we are still not there, which is actually the MyDoc2022 baseline detector, and it was run on some data set that we, or some slide that we got from the UMC Utrecht. And it also shows breast cancer, of course, and we can see that outside of the region of interest, which would be maybe here, we can find lots and lots of false detections. And I will just briefly zoom in here to see that these come from some, like staining artifacts or some other artifacts, which are very similar in morphology compared to like the actual mitotic figures, but of course are nowhere near that in like stain and so on, and color. And of course, like since we have such a high diversity in color and so on, what the model kind of learned was to ignore that. And this is also what you see in these detection results. So to finally really conclude with my take home. So the impact of the lab environment and the species on the domain shift might be overrated. The dataset size matters by a lot. The dataset diversity matters even more. So concluding, to achieve robust deep learning performance, the dataset diversity is really what you want and what you need. And the impact of domain shift is largely dependent actually on the pathology task. So we had a couple of in the presentation now, and in our investigations, the species itself had actually the least impact. This means that the solution transfer from veterinary histopathology datasets to humans seems actually really viable. Right, so a little bit too long, my talk. Still hope that it was not boring. So I have some time for questions if time allows.
Thanks for the very interesting talk, Mark. I think we could have as one question. Okay.
Thank you, Mark. Can you hear me okay?
Yes, I can hear you, Nasir.
Okay, great. Well, thank you very much for an amazing talk, and thank you again for organizing two wonderful contests, which our team has really enjoyed for obvious reasons. My question is, your most of your talk and the two challenges that you have organized, they have been focused on object detection. Do you have any view on the effects of domain shift on whole slide image level classification?
Yeah. So yes, so we have also some preliminary data on that. I mean, like on whole slide images, you will typically go for approaches like weakly supervised learning approaches and so on. I think it like very much all depends on the stability of your features. What we have seen, and this is only like this is just an impression for now, is that actually some pre-trained like self-supervised pre-trained algorithms might actually be stronger dependent on like color cues, as you might expect. On the other hand, like feature extractors like, for example, the ImageNet based ones are much more robust than we expected initially. So we just had also some kind of molecular profiling run on whole slide images, and we did that like using different scanners, and it was surprisingly robust, without any doing kind of, like adversarial training or augmentation or whatnot.
Thank you.